Now, before moving to fertilization, let's have a quick overview about the structures of the female gamete. The female gamete, which may be also called as secondary oocyte, ovum, or mature oocyte or oocyte by some authorities. The female gamete is very large cell. The nucleus of the female gamete is not visible because the nuclear membrane dissolves for the second mitotic division. Spindle and chromosome attached to it in equatorial plane is seen, which suggests the metaphase stage. This female gamete is surrounded by the layer which is called as zona pellucida. This zona pellucida is in turn surrounded by the cells of corona radiata. A distinct space is present between the cell membrane, which is called as vitelline membrane, and that of zona pellucida, which is called as perivitelline space. It consists of the first polar body. So now, talking about fertilization. What is fertilization? Fertilization is the fusion of male and female gamete. What is the common site of fertilization? The site of fertilization is the ampulla of fallopian tube. While discussing about the fertilization, let's discuss it under the following subheading, which includes approximation of gamete, which we further deal about the migration of sperm and transfer of oocyte. Similarly, fusion of gametes and result of fertilization. Approximation of gamete. Now, in approximation of gamete, let's discuss about the migration of sperm. During coitus, of male ejaculates about a teaspoonful of the semen containing 200 to 300 million sperm in the female vagina. Why this high number of sperm is deposited? Because of the high rate of sperm mortality. Out of this 200 to 300 million, only 200 to 300 sperm manages to reach to the contact of the female gamete. Now, the journey of sperm from the vagina to the ampulla is promoted by the prostaglandin present in the sperm, which causes the powerful contraction. Similarly, this powerful contraction is substantiated by the oxytocin released by the neurohypophysis. Uterine contraction creates the negative power. This negative power causes the sucking of the sperm from the vagina into the uterine cavity. The movement of sperm from the cervix to the isthmus of the uterine tube occurs primarily by the unpropulsive movement, that is whip cord-like movement of the sperm, plus the fluid current created by the uterine cavity. It takes about 2 to 7 hours for the sperm to reach from the cervix to the ampulla of the uterine tube. Now, talking about the transport of an oocyte, just before the fertilization, the fimbriated end of the fallopian tube become closely applied to the surface of the ovary, and its finger-like projection start moving back and forth like a sweeping action over its ovarian surface. As the secondary oocyte is released or discharged, this secondary oocyte is released in the peritoneal cavity. Now, the fimbriated end of the fallopian tube, it enters into the fallopian tube. The oocyte reaches the ampullary end of the tube due to the movement of the cilia and contraction of the muscle of the uterine tube. Now, what is the time for the viability of gametes? The time for the viability of female gametes is 24 hours after fertilization, and that of male gamete is 48 hours in the female genital tract. Now, talking about the fertilization barrier. What is fertilization barrier? Fertilization barrier, the female gamete is protected by the three barriers from superficial to deep. The first barrier is corona radiata. Let's recall about the different layers of the female gamete. It was first barrier is corona radiata, second barrier is zona pellucida, which is made of the glycoproteins like ZP1, ZP2 and ZP3. Now, third barrier is vitelline membrane of the oocyte. So, what is zona? In this, we are going to discuss more detail about the zona pellucida. What is zona pellucida? Zona pellucida is a thick transparent membrane of the three glycoproteins ZP1, ZP2 and ZP3 surrounding the ovum. It is the extracellular matrix which is formed by the secretion from oocyte and zona granulosa cell. So, what is the function of zona pellucida? The function of zona pellucida, it protects the oocyte and early embryo during the development. Similarly, it allows diffusion of the uterine secretion for nourishment of the blastomere, and it regulates the interaction between the oocyte and the sperm around it during the fertilization. The ZP3 protein is responsible for the acrosomal reaction. So what is acrosomal reaction? We are coming up with it in the next slide. So it is also responsible for the zonal reaction, and it also prevents ectopic implantation of blastocysts, which is most important. We know zona pellucida is a protective covering. If there is not if the zona pellucida is not present there, then the trophoblast will have highly sticky nature. It may get implanted into the fallopian tube. So due to the zona pellucida, it protects the ectopic implantation, that is prevent the implantation of the blastocyst at a site other than the normal site. So before talking about the fertilization, let's discuss about the capacitation. What is capacitation? Capacitation is the process of conditioning of a sperm. What is conditioning of a sperm? That is, it is the final step of maturation of a sperm. It makes the sperm competent to fertilize an oocyte. What is the time of capacitation? The time of capacitation is after ejaculation of a sperm into the female genital tract. It lasts for about 7 to 10 hours. The glycoprotein and seminal plasma of the sperm in the acrosomal region is removed. Sperm tail start beating more vigorously. How it start beating more vigorously? Due to the calcium channel entry of the calcium ion. Now what is the significance of capacitation? Capacitation of a sperm, it is a prerequisite for the fertilization. So because if the sperm are not capacitated, then it won't be able to fertilize the sperm. Only capacitated sperm undergoes acrosomal reaction and fertilize the ovum. What is acrosomal reaction? Acrosomal reaction occurs when the capacitated sperm comes in contact with the zona pellucida. This reaction leads to the release of enzymes hyaluronidase and acrosin from the acrosomal cap. The antigenic coating of the sperm initiate an immunological reaction between the oocyte fertilizing and sperm's anti-fertilizations. Talking about the stage of fertilization, the first stage of fertilization includes penetration of corona radiata. The second stage includes penetration of zona pellucida. The third stage includes the penetration of vitelline membrane. We can see here these three are the fertilization barrier. So this fertilization barrier must be penetrated first. Now the third step is the formation of female pronucleus. Fourth step is the formation of male pronucleus, and the sixth stage is the formation of zygote. So what happens in the penetration of the corona radiata? About 300 to 400 sperm penetrate the corona radiata. There is the liberation of hyaluronidase enzyme, which causes the digestion of the chemical substance binding cells of the corona radiata. Now what happens in the penetration of zona pellucida? The contact between the head of a sperm with the glycoprotein of the zona pellucida occurs, which initiates the acrosomal reaction. This acrosomal reaction causes the release of digestive enzymes like acrosin and pepsin-like substance, so which causes the lysis of the zona pellucida and plasma membrane of the head of the sperm. Now this allows the sperm to penetrate through the zona pellucida and reach into the perivitelline space. Once the sperm penetrates the zona pellucida, a change in the properties of the zona pellucida now occurs, which is called a zona reaction, that makes it impermeable to the other sperm. Now talking about the penetration of vitelline membrane, plasma membrane of a sperm head comes in contact with the plasma membrane of oocyte, which causes the break in the oocyte plasma membrane at the site of fusion. Now entry of the head of a sperm into the cytoplasm occurs, leaving behind the discarded body and the tail of the sperm on the oocyte surface. As soon as the head and neck of the sperm enters the oocyte, a calcium wave appears in the cytoplasm of oocyte that makes oocyte membrane impermeable to the other membrane. So the polyspermy is prevented here. Now the fourth step in the fertilization is the formation of female pronucleus. The penetration of oocyte by sperm causes the activation of oocyte, due to which the oocyte completes its second meiotic division, and the mature oocyte and the second polar body is formed. Condensation and arrangement of mature oocyte in the vesicular pattern is done to form the female pronucleus. Now the male pronucleus is also formed. The sperm moves forward to come in close contact with the female pronucleus. Its nucleus becomes swollen and forms the male pronucleus. The morphologically, the male and female pronucleus are indistinguishable. So each chromosome in male and female pronuclei is made up of only one chromatid. The pronuclei grow and replicate their DNA, that is, changes from the haploid to diploid. Now, talking about the formation of gametes, formation of zygote, the male and female pronuclei loses their cell membrane, and the chromosome of the two nuclei mix together to form the diploid. Now, talking about the this is the diagrammatic representation of what I have discussed in the stages of fertilization. First of all, the sperm comes in contact with the surface of the oocyte, or it penetrates the corona radiata, then zona pellucida, then the perivitelline space. This way, image is showing the penetration of the sperm through the zona pellucida. So after the penetration of sperm through the zona pellucida, the head comes in contact with the vitelline membrane. It fuses the head, the side cell membrane of the sperm head, and that vitelline membrane fuses, and now the entry of sperm head and neck occurs, and the leaving behind the body and the tail. After that, the formation of male and female pronuclei. The male nuclei becomes swollen and it is morphologically indistinguishable from the female pronuclei. Similarly, when the sperm enters into the cytoplasm, the head it also completes its second meiotic division and giving rise to the female pronucleus, and we can see here the second polar body. Now, talking about the replication of DNA occurs. Replication of DNA occurs from 1N to 2N. Now, the breaking of the cell membrane of the nuclei and the chromosome mixes up, and cleavage spindle is start, and there is the formation of the female gamete. There occurs the formation of the zygote. The chromosome of the zygote get arranged in the equatorial plane because of for the further division. Now let's discuss about the fertilization illustration, which is this summary of the fertilization which have been taken from the boron physiology. So the sperm cell weaves past follicular cell, binds to the zona pellucida, as arise in after binding to the zona pellucida, a rise in calcium ion concentration inside the sperm cell triggers the exocytosis of the acrosome. Inside the sperm triggers the exocytosis of the acrosome, which contains hydrolytic enzyme. The hydrolytic enzyme contained in the acrosomal cap are released. This enzyme locally dissolves the zona pellucida. The whip-like action of the tail pushes the sperm head towards the oocyte membrane. With the head of the sperm now lying sideways, microvilli on the oocyte surround the sperm head. We can see here. Now the two membrane fuses, and the content of the sperm cell enters the oocyte. Sperm cell membrane remains behind. Now in fifth stage, what happened? The rise in calcium ion concentration inside the oocyte occur, which triggers the cortical reaction, and there is the exocytosis of the granules that previously lay immediately beneath the plasma membrane of the oocyte. The enzyme released lead to the change in the zona pellucida protein, causing the zona pellucida to stiffen or harden, preventing the entry of the other sperm. Now the rise in calcium ion concentration inside the oocyte induces the completion of the second meiotic division and the formation of second polar body which lies next to the first polar body. Now the head of the sperm enlarges to become male pronucleus. The male and female pronuclei fuse. This is all about the summary of the fertilization. So this picture is explaining the same thing. The first stage is the penetration of corona radiata. The second phase is penetration of the zona pellucida, and the third phase is the penetration of the vitelline membrane. This figure is also saying the same thing, which is taken from the most developing embryo. First, second and third phase. First, sperm containing sperm nucleus containing chromosome, acrosome containing enzymes, and the second step is penetration of the zona pellucida, and the third step is the formation penetration of zona pellucida, penetration of the corona radiata, then zona pellucida, and after that there will be the release of sperm in the cytoplasm oocyte without its plasma membrane occurs. Now, what is the result of the fertilization? The result of the fertilization is that the most important result is that it causes the completion of the second meiotic division of the female gamete. It also causes restoration of the diploid number of chromosome, one n number of chromosome from the male pronuclei and one n number of chromosome from the female pronuclei, which fuses to form the diploid. Similarly, the determination of the chromosomal sex of the new individual is also possible due to the fertilization. So if X chromosome is fused with the Y chromosome, it gives rise to the male zygote, whereas when X chromosome from the male and X chromosome from the female fuses, then it gives rise to the female zygote. Now, the initiation of cleavage is also occur. Fertilization stimulates the zygote to undergo rapid mitotic division, which is called as cleavage. Similarly, the variation of human species is also due to the result of the fertilization. Now, after the fertilization occur, now the beginning of the development of the new offspring has occurred. So how the beginning of the development occurs? In this we are going to discuss about the cleavage and blastocyst formation. What is cleavage? Cleavage is the repeated mitotic division of the zygote into the smaller unit. There is the increased number of cells, which is called as occurs the increase in the number of cells, which is called as blastomeres. So what happens? The zygote undergo division to form two cell stage. This two cell stage, and we can see here the zona pellucida is still intact here. It will be here, it will till the time up to the implantation. Now this zygote divides to form the two cell stage, of which one cell is larger and other cell is smaller. The larger cell divides first, giving rise to the the larger cell divide first to give rise to the three cell stage. The smaller cell divides next to give rise to the four cell stage. This division continues to form the this 4 cell divides to form 8 cell stage, which further divides to form the 16 cell stage. This 16 cell stage resemble the mulberry fruit, so it is termed as the morula. In morula, the blastomere are very small and contain very little amount of cytoplasm to survive. Now, talking about the blastocyst formation, the morula enters into the uterine cavity about the 4 days after the fertilization. The endometrial fluid penetrates the morula and enters into the intercellular spaces of the morula. So what happens? The intercellular space, there will be the formation of intercellular space. This intercellular space, we can see here, it is morula, and there will be there occurs the penetration of the endometrial fluid into the intercellular space. This intercellular space, a small intercellular space, fuses to form a large fluid space, which is called as blastocyst cavity. So the blastocele, which is called as blastocyst cavity or blastocele, the blastocele is filled with the fluid rich in nutrients, which is secreted by the endometrium of the uterus, which is called as uterine milk. Now, the cell of the blastocyst gets arranged in the two groups, which is called as inner cell mass. About 6 to 8 cell gets arranged into the inner cell mass, which is called as embryoblast. It gives rise to the embryonic disc, and outer cell mass, which gives rise to trophoblast. Trophoblast is further divided into cytotrophoblast and syncytotrophoblast. We will be discussing it in the detail in the coming days. So the enlargement of the cavity occurs. So outer cell forming trophoblast flattened, and inner cell mass embryoblast becomes compact and is attached to the one pole. So what is hatching of the blastocyst? What is hatching of the blastocyst? The hatching of as the blastocyst increase in the size, the zona pellucida, which is surrounding the blastocyst, gradually disintegrate and disappear. The shedding of the zona pellucida leads to the hatching of the blastocyst. Now this blastocyst is ready to attach on the endometrium at its embryonic pole. So what is the fate of the blastocyst? The fate of the blastocyst is 3. So what is that? Embryoblast. What is embryoblast? Which comes from the inner cell mass. It gives rise to the embryo. Similarly, trophoblast. What is that trophoblast? It is the outer cell, which is a outer flattened cell of the blastocyst, which gives rise to the placenta and is responsible for the providing nutrients to the embryo. Similarly, blastocele, the blastocele cavity, which gives rise to the or which forms yolk sac. This is the summary of the how different stages occurs. Similarly, the secondary oocyte are released. The secondary oocyte then the secondary oocyte moves into the ampulla of the fallopian tube, where the fertilization with the sperm occurs, and there occurs the time of DNA duplication occurs. After that, the cleavage starts after the 30 hours. Now this cleavage starts and the 16 cell stage is formed, which is called as morula. At the 7th day, similarly morula is formed. At the 4.5 days to 5 days, the blastocyst is formed, and 5.5 days to 6 days, the implantation occurs. Initiation of implantation occurs in the first week. Now talking about the clinical correlation, the clinical correlation is that hydatidiform vesicular mole. The hydatidiform it is the abnormal development of the trophoblastic cell following fertilization of an empty oocyte. This empty oocyte is fertilized either by a single sperm, which is called as monospermic mole, or it is fertilized by the double sperm or two sperm, which is called as dispermic mole. We are going to discuss, we will be dealing it in detail in the coming days. So what now talking about the implantation, what till we have discussed is that female sexual cycle, how the oocyte is released, how the sperm is transferred, and the stages of the fertilization. So what is the different stage of approximation of gamete? Approximation of gamete. After that, what is that? In approximation of gamete, we have discussed about the transport of sperm, similarly transport of an oocyte, then now fertilization and the phases of the different phases of the fertilization. After fertilization, the blastocyst reaches into the uterine cavity about 5 days or 4 to 5 days. Now this process, after that implantation occurs. What is implantation? Implantation is a process by which moving blastocyst is first attached to the epithelial lining of the endometrium and then gets embedded into the uterine endometrium. The implantation which occurs in women is the interstitial implantation, that is the oocyte enters deep inside the tissue of the endometrium, not it remains superficially. Now the site of the implantation is upper part of the posterior wall of the uterine cavity. So talking about the what are the different events or timeline in day 5, the blastocyst surrounded by the zona pellucida enters into the uterine cavity. Now hatching of blastocyst occurs because enlargement of the blastocyst causes disintegration and disappearance of the zona pellucida, which makes it the trophoblast underlying the zona pellucida is highly sticky in nature. This trophoblast bind on the day 7 with the epithelial lining of the uterine endometrium. So actually implantation is the event of the second week, but the initiation of the implantation, that is binding of the trophoblast with the epithelial lining of the uterine endometrium, occurs in the day 7, which is in the first week. So we have discussed a little bit about the implantation in the first week itself. We will be discussing it in detail in the event of the second week of the development. So what are the abnormal site of the implantation? We have already discussed. The normal site of implantation is upper part of the posterior wall of the uterine cavity. So what is the abnormal site of the implantation? The abnormal site of implantation is within the uterus or outside the uterus. Within the uterus, it can be lower uterine segment near the internal os. We can see here near the internal os of the orifice os of the uterus, which is called as placenta previa. Similarly, outside the uterus, if it occurs outside the uterus, it is called as the ectopic pregnancy. It may occur in the uterine tube, which may be either at the ampulla, infundibulum, ampulla, interstitial part, etc. Similarly, in the abdominal cavity, it can be in the pouch of Douglas or ovary itself. Now summarizing all the event of the fertilization and implantation, the different layer of the female sexual different stage of the female sexual cycle, we are going to discuss about the after the ovarian cycle, the secondary oocyte is released at the time of ovulation, that is 14th day. This secondary oocyte is cast by the fumariated end of the fallopian tubes and it enters into the fallopian tube. The ciliary movement of the lining of the fallopian tube or uterine tube causes the movement of secondary oocyte from the or female movement of female gamete from the infundibulum into the ampullary region. The in ampullary region of the fallopian tube, it gets fertilized by the sperm, and there will be there occurs the formation of zygote. This zygote undergo cleavage to form 2-celled stage, then 4-celled stage, then 8-celled stage, and then morula. This morula is get filled by the endometrial fluid. There occurs the formation of intercellular space. The intercellular space is coalesces to form blastocele, and the one group of cell gets arranged in the one pole, which is called as blastocele, which leads to the formation of the blastocyst. In blastocyst, what occurs? Some cell, that is 6 to 8 cell, gets arranged in the one pole, which gives rise to the inner cell mass. This inner cell mass gives rise to the embryo, and similarly the flattened cell, which is called as trophoblast, this trophoblast gives rise to the placenta, which is responsible for the nutrition. Similarly, the blastocele cavity, this blastocele cavity is give rise to the yolk sac, and after that in the posterior wall of the uterus it gets implanted. What are the different site of abnormal site of implantation? It may be inside the uterus or outside the uterus. What are the inside the uterus? It may be near the internal os of the lower uterine segment. Similarly, it may be into the ampullary region and infundibular region or interstitial part of the fallopian tube. It may be into the ovary also, or it may also occur into the pouch of Douglas, that is the peritoneal fold between the uterus and the sigmoid colon. So this is all about the summary of the ovarian cycle, fertilization, and the development of women in the first week. So these are my references. Thank you for your patience.